no human being has caught it in New York City for at least a century, but still, there it was, researchers said, in places touched by hundreds, maybe thousands, of people every day. After swabbing more than 400 subway stations for all kinds of microorganisms, researchers at Weill Cornell Medical College reported this week that they had found evidence at points across the city of bubonic plague, the Black Death that ravaged 14th century Europe. This was hardly surprising. Everyone who rides the subway knows it is teeming with rats, which in the right environment can be infested with fleas that carry plague. Medieval quantities of rats. It also is scary. Could a subway turnstile be all that is standing between New Yorkers and a nearly forgotten disease? Probably not. On Friday, the day after the study's release, the city's health department questioned whether the researchers had turned up the plague at all. "We don't know what bacteria they found, but it's definitely not the plague," Levi Fishman, a spokesman for the department, said. The study's authors hedged a bit, allowing that what they thought was the plague could actually have been a different bacterium. Or it could have been a plague. "We're saying there's evidence for these things," Christopher E. Mason, a geneticist and the lead author of the study, said Friday. But no one should worry. The study, published on Thursday in the online journal Cell Systems, was intended to create the kind of microbial map of the subway system, collecting and analyzing the DNA of the humans, insects, bacteria, plants, viruses and other types of life that comprise the subterranean melting pot. Most of it was harmless. But the researchers found fragments of DNA associated with two not-so-harmless pathogens, anthrax, which Dr. Mason guessed might have originated from livestock and has not been seen in New York in several years, and plague. None of those fragments were in live samples, meaning they could not have infected anyone touching them. If it was the plague they found, the discovery is quite a breakthrough, given how long it has been since the disease was present in the city. Mr. Fishman said the complex ecology that does not occur in New York, much less on the subway, is needed for humans to contract plague. The bubonic plague did find its way to New York in 2002, traveling with two visitors from a ranch in New Mexico, where the disease is endemic in flea-bitten wild animals like prairie dogs. Dr. Mason said it was possible other tourists had carried the plague onto the subway, even if had not been detected before. The United States sees an average of 7 cases of bubonic plague a year, concentrated in the rural West, according to the Centers for Disease Control and Prevention. Dr. Mason said the fragments of DNA associated with the bubonic plague were found at 3 stations in disparate parts of the city, on a garbage can at the 103rd Street station on the number 6 line in Manhattan, the stairway railing at the 111th Street station of the 8th line in Queens, and another railing at the Winthrop Street station of the number 2 and number 5 lines in Brooklyn. Dr. Mason said his team wondered how rats were able to balance on railings. "I'm not an expert on rodent physiology," he acknowledged, "so we had these funny jokes about acrobatic rodents." But he said it was also possible that the puff of air created by moving trains kicked up the DNA on the ground, so it comes from the aerodynamics of the subway. Mr. Fishman conceded that rats have been known to frequent the NYC subway. But he said, no infected rat has ever been found in NYC. What the researchers probably found, he said, was bacteria from an unknown species or from organisms that happened to share some gene sequences with the plague bacterium, Yersinia pestis, or Y. pestis for short. Dr. Mason agreed that his team might have detected some other bacterium masquerading as plague. But it is also possible, he said, that the plague was actually there. "To truly know if it is the plague, you have to take a piece and infect someone," he said. To prove their good faith, he said, the researchers plan to post a more extensive explanation of their data online, so that literally the middle school student could do some of the analysis. Assuming it was plague, Dr. Mason had a theory for why it had not wiped out New York City. The strain of plague from the Middle Ages was a particularly bad strain, he said. New York City today, he also noted, by comparison, even though some people might complain, it's a far cleaner environment than the Middle Ages. What's next for Dr. Mason and his pathogen mapping team? First they will see if they can link the suspected plague DNA to the rats, he said. Then they will try to refine their DNA matching to look for 2 other notorious urban pests, cockroaches and bedbugs.